However, these are essential in pregnant women, lactating mothers, and in growing children. Okay, these are required but not synthesized by our body sufficiently during growth. These are required mainly by requirement for women, lactating mothers, and in growing child. These amino acids have a important role as a growth promoting nutrients. Okay, as a growth promoting nutrients, these are having highly importance. Examples are arginine and histidine fall under this category. The examples of semi-essential amino acids are arginine and histidine. Next, coming to non-essential amino acids. These amino acids can be synthesized by human body from other materials through a series of biochemical reactions. It is therefore not necessary that they should be consumed through diet. As these are synthesized by our own human body, these are need not to be taken externally. Those type of amino acids are non-essential amino acids. Next, coming to some of the important amino acids names and their abbreviation code, and this is a structure of the amino acid. See, everyone contains a parent atom, and based on different atoms attached to the side chains, see, side chains, these are given different names. Okay, see, this is the parent atom which is present in all structures, which is a parent atom, and based on the substituents of these parent chains, these are being given different names. See, these are the important amino acids. These all are neutral amino acids. These are neutral amino acids with polar but unionized side chains. Unionized side chains, non-polar side chains. Neutral, both are neutral amino acids with non-polar side chains, and here are polar side chains. Okay, these are the important amino acids, namely glycine, alanine, valine, leucine, isoleucine, methionine, pyrroline, phenylalanine, and tryptophan. Next, these are asparagine and glutamine. Next, coming to stereochemistry of amino acids. Stereochemistry of amino acids. Mainly, except glycine, all amino acids contain a stereo center. As I discussed in previous classes, what do you mean by a stereo center? That is a chiral center. A carbon attached to 4 different functional groups is known as a chiral carbon, hence thus optically active. A compound containing chiral center is supposed to be optically active, except in glycine. Remaining all amino acids contain at least one stereo center, hence they are optically active. See the difference. In glycine, due to the presence of 2 identical atoms on carbon, this carbon cannot be taken as a chiral center, whereas in other amino acids there is a stereo center because this is attached to an R group where hydrogen is replaced by R. Next, coming to the amino acids may be dextrorotatory or levorotatory depending upon the pH of the solution and nature of the side chain. Except glycine, rest of the amino acids occurs in D form and L form. Okay, this is a D amino acids. This is an L amino acid. Okay, this is D, dextro. This is L. See, these are placed here. Nitrogen is present here. So, based on rotation, these are given D forms and L forms. Next, coming to natural amino acids occur in L form. Some of the important concepts regarding our stereochemistry aspects of amino acids. Natural amino acids, all of the natural amino acids occurs in L form, that is levorotatory. See, levorotatory 1, 2, 3 and like this. So, left side or rotation is seen like this. See, from 1 to 1 to interchange. So, like this. The natural proteins obtained from animals and plants in general contains L amino acids. Bacteria contains D amino acids. The L amino acids exhibits S configuration. The L amino acids exhibit S configuration. See this direction. So S configuration. Exceptions to this are cysteine and cysteine. Cysteine and cysteine. When sulfur is present in the side chain R and thus in the assigning the configuration, the side chain gets higher priority over COOH groups. Hence, the explanation is given in these structures. Okay, except to the cysteine and cysteine where sulfur is present in the side chain. See, sulfur is present in this side chain and thus assign the R configuration. Hence, thus R configuration is assigned. Rest all are S configuration. Only exception is for cysteine and cysteine, which contains sulfur in the side chain. Hence, they show R configuration, whereas remaining all the amino acids, that is natural amino acids, show L form and S configuration. Next, coming to the important methods of synthesis of amino acids. The important methods for the synthesis of amino acids are: first one, Gabriel-Thalmide synthesis. Gabriel-Thalmide synthesis. So, thalamide is converted to its reactive salt by reaction with ethanolic KOH. Okay, see in this reaction, thalamide is reacted with ethanolic KOH. As a result, H plus is substituted by K plus, results in the formation of a highly reactive intermediate, that is potassium thalamide, which is highly reactive. The potassium salt of thalamide on reaction with alpha halo acids. Okay, the carbon that is attached to functional group bearing carbon is called as alpha carbon. This is the alpha carbon and this is a functional group bearing carbon. This is the alpha carbon. Alpha halo, as the alpha carbon is attached to chlorine, such kind of compounds are known as alpha halo acids. Followed by hydrolysis results in the formation of amino acids. Okay, this potassium thalamide reacts whereas KCl get eliminated and this carbon gets attached to the nitrogen. Hence, this intermediate is formed. Followed by hydrolysis. As we all know that ester on hydrolysis give alcohol and acid. Same, acid and alcohols are formed, as this is converted into phthalic acid and this alpha amino acids are synthesized. Next, coming to Streaker synthesis. It is another important method for the synthesis of amino acids. An aldehyde on reacting with potassium cyanide in the presence of ammonia or ammonium chloride results in the formation of a alpha amino nitrile. This is alpha. The carbon attached to functional group is alpha carbon. Alpha group is attached to amino group. Alpha amino nitrile. Hydrolysis of alpha amino nitrile in acidic medium yields an alpha amino carboxylic acid. The overall reaction is known as streaker synthesis, which basically involves the conversion of an aldehyde to alpha amino acid having one more carbon atom than that present in the aldehyde. For example, see the aldehyde taken is ethanol, which contains two carbon atoms, and the resulting alpha amino acid contains three carbon atoms as stated in above lines. Okay, these are the two important methods for the synthesis of amino acids. Next, coming to another important topic in our syllabus, that is high throughput screening. Okay, high throughput screening. It is also known shortly it is considered as HTS. Okay, high throughput screening. What do you meant by high throughput screening? It is an important topic in our CAD syllabus, that is computer aided drug design. In computer aided drug design, we use high throughput screening for the analysis of large number of ligands to a specific target in limited or very less amount of time. Okay, well, coming to what do you meant by high throughput screening? It is a process where a large number of diversified chemical compounds are tested per hour against a particular targets to identify the hits. Okay, as I discussed earlier, high throughput screening is nothing but it is a process where a large number of diversified chemical compounds are tested per hour against a particular target. That the target molecule is nothing but the site where the action has to be shown against a particular target to identify the hits. Okay. Next, what do you meant by hits? These are small subset of compounds that display desired property. That is, we will be testing a large number of compounds. All the compounds, there is no compulsory that all the compounds should show certain action. So, only a limited amount of compounds in the huge number of taken compounds will show the desired activity. Such compounds are considered as hits. Next, coming to high throughput screening. It generally comprises of a robotic handling system, a liquid handling system, a sensor system, and a control software. First one is robotic handling system. It consists of one or more robots. Functions to transport assay plates from one station to another, mixing, incubation, and finally detection of sample and reagent. The major function of robotic handling system is to transfer the assay plates from one station to another station, mixing them, incubating them, and finally detecting the sample. These robots can test up to 1 lakh compounds per day. 1 lakh compounds per day. If it exceeds 1 lakh compounds per day, then it is regarded as ultra high throughput screening. Okay. Next, coming to liquid handling system. What does it do? It used to serve, it is serves as to fill the wells in the microplates. This liquid handling system is mainly used to fill the wells in the microplates. Next, coming to sensor systems. These sensor systems, they evaluate the sample after incubation efficiently and with high sensitivity. These are used for evaluation of the sample. Sensor systems are mainly used for effective evaluating the samples with high sensitivity. Next, coming to control softwares. It choreographs the whole process, ensuring efficiency within the process and repeatability between the process. Okay, control softwares. This control software, it choreographs the whole process, ensuring efficiency within the process and repeatability between the process. These are the four important components of components of high throughput screening, which is helping us to identify the hit molecules. By knowing the hit molecules, we can undergo several derivatization techniques such that the hit molecules are synthesized synthetically, and various derivatives of that synthesized molecules have been synthesized, which may have less or more activity as that of the hit molecules. Okay, thank you.